Influenza A viruses are significant pathogens that are always on the move. Every year they cause seasonal outbreaks and occasionally cause pandemics, wreaking havoc on public health. At the core of their success is their ability to invade and spread between the cells of an infected individual. Once inside, the battle begins, a race between the virus and the immune system. The virus hijacks cells, forcing them to churn out copies in rapid succession. Our body, sensing this hostile takeover, mounts a defense aiming to neutralize any virus breaking free. But here's the twist. Imagine the virus as a bank robber, cornered by the authorities that is our immune response. Logic would say it's trapped. But just like in the movies, there's always a way out. Even when surrounded, influenza A viruses don't need to burst out of the cell to spread. Instead, they can perform their own great escape, tunneling between cells, bypassing the immune response. In this process referred to as direct cell-to-cell spread, the virus finds hidden pathways between neighbouring cells, continuing its spread, avoiding capture. But how do they do this? As part of the takeover, influenza viruses don't just use the machinery of the cell to make more copies, but it uses the cell itself to essentially inject copies of the viral genome into neighbouring cells, initiating a new infection. The virus does this by triggering the cell to produce tunneling nanotubes, or TNTs for short. TNTs are thin projections of the cell membrane that extend out and touch neighbouring cells, often at impressive distances. This forms a highway for the virus to go on the run and find safe harbour in the newly contacted cell. Whilst it was known that influenza A viruses could induce the formation of TNTs in the lab, it was not known how they were doing this. Furthermore, we didn't know if these structures could form in the lungs where influenza A viruses infect, and whether they are used by different influenza viruses to spread infection. Using super-resolution fluorescence microscopy, we visualized and quantified TNT-like structures both in cultured cells and within the lungs of infected mice, identifying conditions under which these intercellular connections emerge. Additionally, we used fluorescently labeled viral proteins to determine the efficiency by which different influenza viruses can spread directly between cells when virus particle release is inhibited with an antiviral drug. This provides key insights into non-canonical routes of viral dissemination. To uncover how influenza A virus infection triggers tunneling nanotube formation, we systematically investigated key viral and host factors driving this process. Like piecing together a forensic trio, we tracked the clues influenza viruses leaves behind, examining how viral replication, virion budding, and host cell responses contribute to TNT induction. In our recent publication, we show for the first time TNT-like structures emerging from individual cells within the lungs of influenza virus-infected mice. Additionally, we show that influenza viruses can still spread efficiently between cells, even when virus particle release was blocked using an antiviral drug, revealing that alternative pathways for viral dissemination can be readily accessed by these viruses. Our findings show that TNT-like structure formation is a common feature of influenza A virus infection, but is not triggered by secreted immune signaling molecules or membrane alterations linked to the budding of different virions. Instead, TNT induction relied on active viral replication within the infected cell, combined with the activation of programmed cell death known as apoptosis. Interestingly, when we inhibited apoptosis, we greatly reduced the ability of influenza viruses to spread directly between distant cells, indicating that TNT-like structures triggered by influenza virus-induced cell death could then successfully deliver infection to its neighbours. Our findings expand our understanding of how direct cell-to-cell spread contributes to the within-host dissemination of influenza A viruses. By demonstrating the presence of TNT-like structures in influenza A virus-infected lungs, we reveal for the first time that these structures form not only at infection sites, but also within normal lung epithelium. This discovery suggests that TNTs may play a broader role in both health and disease, emerging even in tissues and organs previously considered challenging environments for fragile nanotube formation. Moreover, our investigation into how influenza virus triggers TNT-like structures and the efficiency with which the virus spreads directly between cells provides deeper insights into the mechanisms shaping infection dynamics. By understanding how influenza virus interacts with its host, we can begin to explore strategies to block its spread at the cellular level. Uncovering these covert pathways of viral transmission offers a crucial opportunity to disrupt even the virus's most elusive movements.